At the start of elongation, this is what our DNA looks like. We have our single-stranded binding proteins in green, helicase in orange, and gyrase in purple attached to the DNA. The very first protein I'll introduce is primase, which will read the template strand in the 3' to 5' direction and will synthesize around 10 RNA nucleotides in the 5' to 3' direction. I'm drawing only around 4 on the screen just to make it simpler. So we're thinking, why are we laying down RNA when we're making DNA? And the reason is because DNA polymerase 3 needs to have a 3' OH end to build off of. So now that we have a 3' OH end to build off of, DNA polymerase 3 comes in and reads the template strand in the 3' to 5' direction and will synthesize DNA in the 5' to 3' direction. And this will also happen in the bottom strand as we see here. Now moving on to the next picture, more DNA has unwound, and in the leading strand, DNA polymerase 3 will continue reading the template strand in the 3' to 5' direction and will synthesize DNA in the 5' to 3' direction. But for the lagging strand, primase needs to come down again to the next replication fork and lay down another RNA primer with a 3' OH end to build off of. Only until then will DNA polymerase 3 read the template strand in the 3' to 5' direction and synthesize DNA in the 5' to 3' direction. This will happen multiple times, and these stretches of RNA plus DNA are called Okazaki fragments. Note that we are making DNA and not DNA with fragments of RNA in it. So DNA polymerase type 1 will come in and remove the RNA primers in the 5' to 3' direction, read the template strand in the 3' to 5' direction, and synthesize DNA in the 5' to 3' direction. When removing the RNA primers in the lagging strand, though, there are little gaps, and DNA ligase will come in on that lagging strand and fuse the DNA ends together, so everything is perfectly connected and continuous. As for proofreading, DNA polymerase type 3 and DNA polymerase 1 will proofread in the 3' to 5' direction, and if it's incorrect, use a 3' to 5' exonuclease activity to cut it out and put in the correct complementary nucleotide.